Kate Adamala's team just broke a rule biologists called unbreakable. Their synthetic cell runs on a genome shrunk below the 113 kbp size scientists swore was the floor for life. This isn't just a lab curiosity, it's a significant step toward what many in the field call the holy grail of synthetic biology, creating life from non-living components. This achievement could fundamentally redefine what we understand as life and open a new branch of biotechnology whose uses are still coming into focus. But the story of how this groundbreaking cell was built is a fascinating series of clever hacks and innovative solutions, all designed to circumvent the fundamental limits of biology as we know it. First, let's be clear about what this creation actually is. It's been dubbed SpudCell, and it's not truly alive in the conventional sense. Instead, you can think of it as a highly organized bag of molecules, specifically a simple lipid membrane called a liposome, which is carefully filled with just enough molecular machinery to function in a very basic way. The true innovation lies in its minimalist genome, which is composed of just 7 small units of DNA, known as plasmids. In total, this entire genome consists of just 90 kilobase pairs. For a sense of scale, the human genome, by contrast, has approximately 3 million kilobase pairs. This synthetic cell is, by design, incredibly fragile and highly dependent on its environment. It lacks the internal machinery to make its own proteins or efficiently process its own food sources. As a direct result of this dependency, it's also extremely slow in its processes. Where a natural bacterium like E. coli can divide roughly every 30 minutes, SpudCell requires about 12 hours to complete a single division cycle. This isn't a flaw in its design, rather it's a direct consequence of its minimalist construction. However, that very total dependency is also the precise thing that makes it so incredibly powerful for future applications. Here's the significant payoff for you and for the field of synthetic biology. Kate Adamala herself emphasized this, stating, "I know the full ingredient list of the cell. I know exactly what chemicals, what molecules, at what concentrations." This unparalleled knowledge means it's fully engineerable and predictable. It truly marks the beginning of what Adamala optimistically calls the true age of bioeconomy. This work provides a crucial proof of concept that other scientists can now build upon, adding sturdier systems for metabolism and division. It's the first significant step toward a future of fully engineered biology. But before we fully enter that era, there's one fundamental question that even the experts in the field can't quite agree on. Is this thing actually alive? It undeniably possesses several key characteristics of life. It can grow, it can replicate its DNA, and it can divide. However, it cannot survive independently in the wild, and it lacks the capacity to evolve over many generations without external intervention. One expert commented that it's much closer to being alive than anything else we've created, while another, who even collaborated on founding an institute with Adamala, stated flatly, "I don't think she's created life." This research powerfully demonstrates that the fundamental functions of life don't necessarily require a mysterious magical spark, but it also leaves one question hanging over the whole field. Once you can write the blueprint for life, who decides what gets built next? Stay patched.